Hello, students. So we finished up talking the general stuff about the immune system. Now we're going to begin talking about some specifics, and we're going to look at B cells first, and we're going to look at their steps in activation. So remember, B cells, they start out in the bone marrow, and remember they're going to go somewhere to a lymphoid tissue. Before they do that, don't forget that they have gone through a maturation process. And during that maturation process, they have developed a surface receptor, which is unique. Every individual B cell has its own unique surface receptor. And then they've been tested. They go through that positive and negative selection. And remember, if they fail either one of those tests, then they're destroyed or at least inactivated. And then after they pass both those tests, they're called or said to be immunocompetent. Well, those immunocompetent cells then leave the bone marrow and they go to lymphoid tissues, and they basically just sit there and wait. And what they're waiting for are antigens to pass by. And so remember, if you look at the lymphatic system, we have this fluid which has been out in the tissues, and the fluid travels through the capillaries and then the lymphatic collecting vessels and finally enters a lymph node. And you can see a lymph node in this picture. And then that fluid trickles, it moves around and through all of the little hollow spaces in the lymph node. And as it does so, the fluid moves by those B cells, which are just sitting there. And if there's any antigens in the fluid, the antigens then pass by. And so they pass over the receptors. And so if you look at this, you can see a bunch of B cells here. They all have these different shaped receptors. Remember, each one is unique. But here comes an antigen. Well, B cell A, B, Y, C, Z, and so on, their receptors do not match that antigen, and so they don't do anything. But B cell X, the receptor and the antigen match. And once they match, that cell is said to be sensitized. Now, it can't do anything yet, it's just sensitized. What happens next is that it must go find help. Now, this is not going to happen with any antigen unless it fits into that receptor. Remember, it's like a lock and a key. So here we see three antigens, but this B cell is only going to respond to one of those because the only one that matches its receptor is this one at the bottom. But after it happens, we have this sensitized B cell. And then a sensitized B cell then seeks help. And so we haven't talked about helper cells or T cells yet, but we will. So here's our B cell, and you can see the receptors and you can see the antigen attached. Well, what it does is it's going to use some of that antigen. It's going to pull it inside, and then it's going to hook itself up with this MHC that we talked about, and then go look for help. And so they actually come in close contact and they're sharing this antigen. They both fit. And then the T helper cell, which you see on the left, is going to release hormones. And you can see it says IL-2, 4, and 5. IL stands for interleukins. These are eicosanoids. So back when we talked the endocrine system, we talked about eicosanoids just a little bit. We said that they were local hormones or paracrine secretions. Well, look, these two cells are very close to each other, and so the hormones don't have to travel very far. But those hormones will interact with the B cell, and the B cell will become activated. So here we are again. You can see our B cell, and you can see our T helper cell, and you can see these interleukins and other hormones, and we're going to get an activated B cell. Again, the MHC hooks up with this T cell. Well, the next thing that's going to happen is that particular B cell, none of the others, but that particular B cell will begin to divide. It'll go through mitosis over and over and over again. And what we're going to get are what's called clones because they're all exactly identical. So one cell will form two, two will form four, and then 8, 16, 32, 64, 128, 256, 512, 1024, and so on and so on and so on. And it doesn't take very long for that one cell to create millions of clones. Well, what's going to happen next is those clones can become one of two different things. They can either become plasma cells or they can become memory cells. Let's look at the plasma cells first. So, plasma cells are basically little protein factories. And what they do is they shed their surface receptor. They don't look like B cells anymore, and they begin to produce proteins. And these proteins are called antibodies. And those antibodies are released into circulation. A single plasma cell can produce around 2,000 antibodies per second. And remember, we have millions of these. And so, millions producing 2,000 antibodies per second, the amount of antibody is going to increase tremendously and very quickly. And plasma cells will continue to do that until they wear themselves out. They basically work themselves to death. Again, here is our B cell and hook up with the T cell, and we're going to get activation. And so if we look at that, it looks like this. There's 3 B cells. Remember, the only one that's going to respond is the one in which the antigen fits into the receptor. That B cell is now sensitized. And so if you look to the left, it's going to go find a T helper cell. They're going to hook up. T helper cell is going to release those interleukins, and we're going to get an activated B cell. Our activated B cell is immediately going to divide. And it doesn't just happen once, it happens many, 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 many times. And so we get lots and lots of these clones. And then what happens to the clones is they begin to differentiate. They turn either into plasma cells or memory cells. Plasma cells lose their surface receptor. Look, they don't look like B cells anymore. They have a lot more rough endoplasmic reticulum. They turn into little protein factories, and they start chugging out antibodies at a rate of about 2,000 per second. And it's those antibodies that are going to go out and destroy the antigen. We'll talk about antibodies in just a second. Well, let's also talk about plasma, or the cells that don't become plasma cells. They become memory cells. So memory cells are different. They still look like B cells, but they're very, very long-lived cells. They've already gone through this process, and so they don't have to be activated by a T cell. And so if they ever encounter the antigen again, they remember it. And once they encounter that antigen, they are self activating. And so immediately they begin the process. They don't have to go and look for a T cell. So it's much, much, much faster. So instantly almost, they begin to divide. They produce those clones. And again, clones can turn into either the plasma cells or more memory cells. And so the process happens all over again. So here you can see our original B cell. We went through the whole process, and then we created a memory cell. And then that antigen came along again. It could have been months, could have been years. But as long as there were memory cells still available in the body, we're instantly going to go through this process again. Again, they don't need to be activated, they're self activating. Not only that, but the second exposure creates a much bigger response. So, look, we get more clones, we get more plasma cells, we get more antibody and more memory cells. It happens much faster, and it happens at a much higher degree. So here you can see B cells. Remember, they have released these antibodies as plasma cells, and here you can see antibodies. So let's talk about antibodies themselves. So antibodies are just proteins. They're not alive. They don't do anything except float around, and then when they find an antigen that fits, they bind to the antigen. They don't bind to anything that won't fit into their surface receptor. And their surface receptor is exactly the same as the surface receptor of the B cell. So if it could match the B cell, it matches the antibody. And so if we look at a summary of all that, remember we're going to get production in the bone marrow. The B cells are going to stay in the bone marrow. They kind of go to school there. They're going to develop their receptor. We're going to go through positive and negative selection. We're going to leave the bone marrow. We're going to go to lymphoid tissues. They're just going to sit there and wait until antibodies pass by. Once an antibody fits the receptor, the B cell is sensitized. It goes and seeks help from a helper cell, becomes activated, and clones form. Some of those clones are going to become plasma cells. Some of them are going to become memory cells. And then remember, we're going to make antibodies. So this is what an antibody looks like. So look, it's 4 peptide chains. There are 2 that are long. They're called heavy. And there are 2 that are short. They're called light. And where the ends are is where this unique shape is. Exactly the same shape that we had on the B cell. And that's where we're going to get binding to the antigen. Well, remember, B cells became sensitized, and then they had to find a T helper cell to become activated, and then they had to divide. That takes some time. And so there's a lag period between the antigen challenge and the time that the first antibodies are produced. It's usually about a week, 3 to 7 days, something like that. And then it has to ramp up. And so we don't reach maximum antibody production until about 10 days later. And then antibody levels level off and finally they decline. And so if you look at it, it looks like this blue line. Well, look, if you look at where the first exposure to antigen X was, we can see that about 5 or 6 days, we didn't have any antibodies. And then on about day 5 or 6, we start producing antibodies. And then on about day 10, we reach maximum antibody production. And then it begins to decline as those plasma cells die. But look what happens if you're exposed to the antigen again. So if you look at that second exposure to antigen X, that's on day 28, look how much faster we got antibody production. It usually happens within 24 to 48 hours. And look at the level that you reach. It's about 10 times or so higher than the first time. We get a much bigger response secondary, and then it is maintained for much, much longer. It doesn't decline nearly as quickly. So in general, anyway, we don't get sick the second time, at least not the way we did the first time. You probably have heard of someone having a 24-hour bug. Well, a 24-hour bug is nothing but a bug you've been exposed to previously. And so within 24 hours, you might feel kind of bad or even sick, but usually within 1 to 2 days, you're back to normal again, instead of it being 7 to 10 days like it was the first time. Look at this person, though. Not only were they exposed to antigen X on day 28, they were also exposed to antigen Y, the red line. And Y was a brand new antigen for them. So look, they had to go through the primary immune response to Y, just like we did the primary immune response to X. And so there's this lag time of about 5 or 6 days. It takes about 10 days to reach maximum antibody production, and then it declines very quickly. So this is a good slide to show you why it's important to have a booster shot for a disease. So if you take a vaccination, the first shot that you take is a primary exposure to the antigen. But when you go back for the second shot, look what happens. You get a much bigger response. And so if you notice, most of the COVID vaccines are two vaccines, two shots. And you can see exactly why that is. So that secondary immune response is when you're exposed to the same antigen again. Remember, those memory cells are already activated, and so they respond very quickly. And so we're going to get peak antibody production in about 2 to 3 days. Not only that, but the antibodies tend to be more efficient, and then they last longer in your body. Much longer. They can last for months instead of a few weeks. So what is an antibody? Well, again, it's a protein. Remember, it has these 4 peptide chains that are hooked together, 2 heavy and 2 light. They're sort of glob shaped, and they float around in the plasma of the blood. And so they are part of the globular proteins of the blood. There's a specific part called the gamma globulins. They're also called immunoglobulins, and they're abbreviated IG. Remember, they come from plasma cells in response to a specific antigen, and they can combine with that specific antigen. Well, they're not all exactly the same. There are 5 different types or classes of antibodies. There's IgD, IgM, IgG, IgA, and IgE. An easy way to remember the acronym is to remember the word MADGE, M-A-D-G-E, or if you're a gamer, GAMED, G-A-M-E-D. Those letters do not mean anything. So again, here's our basic structure, and look at it. This structure is called a monomer. It's 1 unit. So this is a single unit, but not all antibodies are just 1 unit. Don't forget, we have these 2 heavy chains and 2 light chains, and where the heavy chains and light chains come together at the end, that's where the antigen binds. That's where the lock is for the antigen key. And if they don't fit together, this antibody will not work. Now, this is truly a little bit more what they look like. But look at this. You can see that antigen binding site. And look at this antigen binding site versus this antigen binding site. Look, they look different. Every single one of them, the antigen binding site, just like on the B cells, they are unique. So whatever the B cell antigen binding site, whatever shape it had, that's what the antibody shapes are going to be coming from it. So this antibody came from a different B cell than this antibody. Remember, there are 5 different types, GAMED. They're all a little different from each other. 3 of them, IgG, IgE, and IgD, are monomers. IgA is a dimer, in other words, 2 units. And IgM is a pentamer, 5 units. Now look, their tails are what are attached to each other, so the binding sites stick out and they're open and free. So look, IgD, IgE, and IgE all have 2 binding sites. IgA, on the other hand, has 4 binding sites, and IgM has 10 binding sites. There are other differences as well. This is what antibodies can do to the antigen. There are basically 6 different ways in which they can destroy or get rid of the antigen. If you look, you can see neutralization. So the antibodies have basically surrounded this virus or maybe a toxin. It's surrounded. It doesn't match the way it's supposed to. It's much bigger than it was. It becomes neutralized or inactivated. They can cause agglutination. So, especially IgM, which has those 10 binding sites, can hook up a lot of different things. When you look at blood types, and we talked about transfusion reactions, this is caused by IgM. So, anti A and anti B are of the type IgM, and we get that clumping or agglutination of the red cells. Another thing that can happen is we can create chains of the antigens and antibodies, and it gets heavy. It becomes so heavy it falls out of solution. This is called precipitation. The other things have to do with inflammation. One of those is it can help complement attach. Remember, complement is that series of proteins that create holes in the membranes of bacteria. And so the bacteria then lyses. So antibody attaches to the cell of the bacterium, and then complement uses it almost like a handle to grab onto so it can attach itself to the bacterium. Another thing that antibodies can do when they surround a particular antigen, they make it easier for white blood cells to phagocytize it. The word for this is opsonization, which literally means to make tasty. So, in other words, it's easier to eat. Another thing they can do is they can increase inflammation, and then they can also, again, help with complement and cell lysis. Here you can see a bunch of antibodies attached to this. Remember, they're not just going to attach in random places, though. They're only going to attach where the antigen fits into their antigen binding site. So if you look at this little graphic, it's kind of fun, but it is a pretty good little short description of what goes on. So the B cell finds an antigen that matches its receptor, goes and finds a T helper cell. T helper cell activates it and begins to divide like crazy. We get plasma cells and memory cells. Plasma cells make antibodies. Antibodies go out and destroy the antigen. One of the ways they can do that is to make tasty or opsonization so that white blood cells can eat them. And then remember, we're also going to make not only plasma cells, but memory cells. And memory cells, again, are really long lived and self activating. Well, because antibodies are proteins and because they're not alive, we don't react to them unless we have whatever it is that they're fits into their binding site. So we can pass antibodies from one person to another. We can create our own antibodies. And so when you create immunity through antibodies, there's actually 4 different types of immunity. There's naturally acquired active immunity. There's naturally acquired passive immunity. And then there's artificially acquired active immunity and artificially acquired passive immunity. So let's look at these just a little bit. So naturally acquired. Naturally acquired means that however you obtain this, it was a natural way. You built those antibodies yourself. They happened in your own body. Nature did it. But you can also get them from somewhere else. You can get artificially acquired. In other words, something weird was going on. So we can have naturally acquired active and naturally acquired passive, artificially acquired active and artificially acquired passive. So, what's an example of those? Well, an example of naturally acquired active is when you just come into contact with something. You touch a doorknob, someone sneezes on you, something like that, but you come into contact with it, produces an infection, and then you make your own antibodies to combat that infection. Naturally acquired passive means you didn't get those antibodies from yourself, you got them from somewhere else. Well, the only natural way to get antibodies from somebody else is what happens with mother and infant and mother and fetus. So a pregnant woman will pass her own antibodies through the placenta. And when that fetus is born, it will have those antibodies and be immune, at least temporarily, to whatever she was immune to. If that mother breastfeeds, she can pass antibodies in breast milk. And so she will continue to give her antibodies to the infant as long as she continues to breastfeed. It's a very natural way to get antibodies. Let's look at artificial. So, artificial means something weird is going on. It's just not something that happens in nature. So, artificially acquired active is what happens when you get a vaccine. It is just not normal to take a needle and inject yourself with something that can make you sick. It is just not normal. Well, now, usually we don't use things that make you sick because we kill them or we weaken them in some way. But still, you're injecting a dead or attenuated pathogen into your own body. And that is just not a normal thing to do. But what happens is once you do that, your immune system will treat it just like you got it from somewhere else, and you'll build antibodies and memory cells to it. And so you're going to have an active immunity. You built those antibodies. Artificially acquired passive immunity is when we pass antibodies from one person to another, but we do it in an artificial way. It's not mother to child, it's not mother to fetus. It's an artificial way. In other words, it's like a transfusion. In other words, you took blood or you took plasma from a friend or someone else or from an animal, and then you inject that into your body. Well, those antibodies, they work. They don't know where they came from, but this is an artificial way of achieving it. So if we go back to COVID again, you probably have heard of convalescent plasma. So convalescent plasma is artificially acquired passive immunity. You've heard of the vaccine. So when you get the vaccine, that's artificially acquired active immunity. If you happen to be one of those people who got sick and you recovered, you have naturally acquired active immunity. If you happen to be a pregnant woman and you got the disease and you got sick, but you recovered and you passed those antibodies to your fetus, that fetus has naturally acquired passive immunity.